The lymphatic system consists of a network of thin-walled vessels that transport lymph from the tissues into the bloodstream. Lymph is interstitial fluid outside of cells, that contains immune cells, lipids, and foreign material. As blood diffuses through the capillaries and into the tissues, some of the fluid leaks out, such as nutrients and small cells. This is where lymph comes from. Lymphatic capillaries are intertwined with regular capillaries and have many openings that allow this interstitial fluid to make its way back into the bloodstream. Lymphatic vessels first drain into lymph nodes, where the lymph is filtered for any foreign substances. These nodes contain lymphoid cells such as T cells and B cells, which work to destroy any antigens in the fluid, mainly with the use of antibodies. The lymph is then returned to the bloodstream via ducts, such as the thoracic duct, which link the circulatory and lymphatic systems. The main role of the lymphatic system is to maintain immunity by filtering lymph for any foreign particles and destroying them by initiating an immune response, which involves B cells and antibodies, as well as other cells such as phagocytes. The tonsils are fleshy lumps of soft tissue located at the back of the mouth, with one on each side. They are a type of secondary lymphoid organ. Primary lymphoid organs are the bone marrow and thymus, and this is where lymph cells like T cells and B cells are made and mature. Secondary lymphoid organs are the site of T cell and B cell activation. Tonsils are one of our first lines of defence and contribute to immunity by trapping foreign particles we may breathe in, like bacteria and viruses. They work to protect the lungs and alimentary canal from harmful pathogens. If a pathogen does make its way into the body, the tonsils have several defence mechanisms in place that help destroy it before infection takes place. The tonsils are covered by a layer of epithelial tissue, and beneath this are epithelial cells that are able to consume bacteria through the act of phagocytosis. These cells then transfer the bacteria to larger phagocytes called macrophages, which present the pathogen to B cells inside the tonsils. A particular B cell will have the correct receptor for the antigen, and once it is detected, the B cell will clone many times and produce antibodies in order to destroy it. Some of these cells remain as memory cells, helping to quicken the body's immune response to that particular antigen should it be exposed again, and thus aiding overall immunity.
The lymph nodes are another important secondary lymph organ of the lymphatic system and play an important role in immunity. They are ovoid in shape with a size range of a few millimetres to approximately 2 centimetres in width and are found throughout the entire body. Lymph nodes are made up of a fibrous capsule, cortex, paracortex, nodules, sinuses, trabeculae, germinal centres, helium, and medulla. These are key sites for white blood cell proliferation, in particular lymphocytes, T cells and B cells, and macrophages. These different cells are produced in different regions of the lymph nodes, with B cells being produced in the germinal centres, T cells in the paracortex, and macrophages in the medulla. These cells help to fight off infection and pathogens by acting as filters. Lymphatic fluid carries dendritic cells containing antigens to lymph nodes via afferent vessels and present the antigens to T cells. These become activated and proliferate, as well as assisting in the proliferation of B cells. Some of the T cells leave the lymph node to find the source of infection in the body and attack the antigens, and B cells with specific receptors bind to the particular antigen and produce antibodies to destroy them. This causes an immune response called reactive lymphadenopathy, swelling of the nodes. Macrophages arrive and use phagocytosis to ingest the antigens, and once the fluid is clear, it passes through the efferent vessel and eventually into the systemic circulatory system. Peyer's patches are also a secondary lymph organ, which help to monitor and maintain a healthy small intestine. They are egg-shaped nodules located throughout the ileum in the mucosal layer and monitor antigens and pathogens travelling through the lumen of the ileum. These patches coordinate an immune response to pathogens and tolerate innocuous microbes and food, acting as a filter like other secondary lymphoid organs. Microfold cells, or M cells, are specialized epithelial cells that line the surface of Peyer's patches and assist in transporting antigens and pathogens from the lumen of the small intestine across the epithelial wall. Inside Peyer's patches are lymphocytes, T and B cells, macrophages, and dendritic cells, assisting in the defense mechanism against harmful antigens. Dendritic cells often sample the lumen of the ileum by using their dendrites to penetrate between epithelial cells. M cells also assist in sampling by phagocytosis of antigens and transcytosis to dendrites. The dendritic cells carrying the antigens travel to the lymphocyte zones of the Peyer's patches, where the T cells activate. These activated T cells initiate activation of B cells, which create antibodies to combat the antigens, with both lymphocytes proliferating. In the occurrence of a breach in the epithelial wall of the mucosal layer, enabling an influx of bacteria, T and B cells proliferate to combat the large influx. Macrophages ingest antigens and neutrophils undergo netosis, forming an extracellular trap and plugging the epithelial wall. Once homeostatic conditions in the Peyer's patch are maintained, neutrophils undergo apoptosis, and macrophages use phagocytosis to remove this waste. The cleared fluid then passes through back into the lymphatic system and eventually into the systemic circulatory system.
Another lymphoid organ is the spleen. The spleen is located in the upper left side of the abdominal cavity underneath the diaphragm. It is a concave-shaped organ sitting on top of the stomach. As a secondary lymphoid organ, its sole purpose is to aid in immunity. Its function changes from that in an unborn child to that in a baby. In an unborn child, its primary purpose is to produce blood and defense cells, whereas after birth, one of its functions is to remove and destroy old and damaged red blood cells. Another function of the spleen after birth is to store macrophages and B lymphocytes until they are required for immunity. The macrophages engulf pathogens through phagocytosis, and the B lymphocytes produce antibodies if required. Platelets and white blood cells are also stored in the spleen. Similarly, the appendix, which was once believed to be a vestigial organ, is also a secondary lymphoid organ and is now said to be essential in maintaining immunity against pathogens. It is a small pouch-like sac of tissue located in the cecum in the lower right abdomen and is only found in some mammals. Recent studies show that the appendix has large amounts of lymphoid tissue, which accumulate in the appendix after birth. The lymphoid tissue aids in the maturation of B lymphocytes and the production of antibodies, as well as producing molecules that help transfer B lymphocytes around the body. As well as having high levels of white blood cells, the appendix also produces and protects probiotic colonies in the gut, which are used to restore intestinal flora in case it is destroyed by an infection. Hence, the appendix is an essential part of the immune system.
We hope this video presented a clear overview of how secondary lymphoid organs like the tonsils, lymph nodes, Peyer's patches, the spleen, and the appendix support the immunology of the body.